Big story out of Boston, Massachusetts, with Boston University doing some type of research on the virus. Should they do it? Why'd they do it? Were they successful in doing it? Was it risky? Let's take a look. So it all starts with this Daily Mail article that came out, what was this, yesterday. Exclusive. This is playing with fire. It could spark a lab-generated pandemic. Ooh, where have I heard that before? Experts blame Boston lab, where scientists have created a new deadly COVID strain with an 80% kill rate. It should probably say in mice, not in people. Researchers added Omicron spike protein to the original Wuhan COVID strain. Well, right off the bat, that doesn't sound too good. Omicron spike is highly mutated and is made most infectious variant ever. 8 in 10 mice infected with the lab-created strain died at the BU lab. That sounds alarming. And of course, the British tabloid, The Mail, specializes in those types of headlines. The Daily Mail continues. The revelation exposes how dangerous virus manipulation research continues to go on in the United States, despite fears similar practices may have started the pandemic, right? You remember that? That it could have come from a lab in China, in Wuhan. That was an unspeakable thing. You couldn't say that until finally one person on the left gave everyone the green light to actually say that. Watch.
So
what do you
takes,
what do you mean by it? Do you mean like
Well, so
perhaps there's a chance that this was created in a lab? There's an investigation.
a chance?
Well, but
oh my
if there is
God.
evidence, I'd love to hear it. I
There's
just don't know.
a novel respiratory coronavirus overtaking Wuhan, China. What do? Oh, you know who we could ask? The Wuhan novel respiratory coronavirus lab. The disease is the same name as the lab.
A leading scientist in the Israeli government said this should be totally forbidden. It's playing with fire. Gain of function research is when viruses are purposely manipulated to be more infectious or deadly. It's thought to be the center of COVID's origin, maybe in a Chinese lab. The lab was, a couple of miles from the whole marketplace. You know the whole story. The practice has been largely restricted in the United States since 2017. That's why we were doing it in China, funded by the federal government. Richard Ebright, chemist at Rutgers, said, the research is a clear example of gain-of-function research. He added, to avoid the next lab-generated pandemic, it is imperative that oversight of enhanced potential pandemic pathogen research be strengthened. That sounds pretty reasonable, but that's not BU's argument in defense. Their first argument in defense was, hey, we're not required to disclose it because we're not directly using money from the federal government. We're indirectly using money, but we're not directly using money, so we don't have to disclose it. That sounds like a dodge. And now on Wednesday morning, according to Yahoo, the federal government is going to investigate the BU COVID controversy experiments and find out what is actually going on. So I actually found this other article at science.org, which I thought was somewhat readable and explained the topic in more depth with maybe not the hype of the Daily Mail. So let's take a look. Was a study that created a hybrid COVID-19 virus too risky? Science looks at the fervor over the experiment at the BU lab. Let's scroll down. And as you scroll down, you find Boston University's second line of defense or argument. Several virologists argued on Twitter that the study is not as alarming as it first appears. For one thing, the hybrid virus was less lethal than the earlier variant modified in the study. So basically, they created something that was less lethal. They also noted that other researchers have published results of similar experiments did not draw similar concerns, and it's not clear the study is very different from other chimeric virus studies that the federal government has exempted from review. Well, just because the federal government has accepted them from review in the past, uh, maybe we should be having some review on these studies, right? As John Stewart said, the name of the virus is the name of the lab, right? What had happened when a penguin kissed a pangolin, or what did he say?
That's just a little too weird, don't you think? And then they ask those scientists, they're like, how did this? So wait a minute, you work at the Wuhan respiratory coronavirus lab. How did this happen? And they're like, a pangolin kissed a turtle.
Okay, that was pretty funny, but let's get back to this science.org article. What did the BU researchers do and why? They took a gene from the Omicron surface protein or spike protein, which COVID uses to enter cells, and added it to the genome of a backbone virus, a variant of COVID from Washington state that was identified after the pandemic emerged from China in early 2020. The objective was to tease apart whether Omicron spike protein explains why it is less pathogenic, causes less disease. The answer could lead to an improved diagnostic test and better ways to manage the disease, preprint authors said. Actually, that makes a lot of sense, like to find like a better test for the virus. Okay, but the simple fact of the matter is COVID's over. Even Joe Biden, who doesn't even know what day it is, told us COVID's over. Excuse me. Somewhat surprisingly, the hybrid virus killed 8 out of 10 mice, where mice were infected with Omicron got sick but did not die. This study suggests the mutations that make Omicron less pathogenic must involve changes in proteins other than the spike protein, authors said. Well, that's pretty darn interesting. But it also says, like, hey, we did gain of function research. We tried to do it, but we failed. We tried to kill all 10 mice, but only 8 of our mice failed. And so, hey, don't blame us. We did try to do gain of function, but it didn't work. So, yeah, we didn't do anything wrong. That's kind of sounds like the explanation to me. And really good explanation from this guy in University College of London. They note that findings made in mice do not also translate in humans. So if the studies is relevant to protecting human health, what does it really care how many mice died? Given such limitations, the arguments for doing this work generally don't feel overwhelmingly convincing to me, tweeted virologist Francois Ballou of University College of London. I'm sure I mispronounced your name. Sorry, Dr. Biologist guy. But good point on your part, which begs the question that obviously if you're doing this in mice, the next step is like, maybe like, or are we going to test this? I don't know. I hope not. If it kills 80% of mice, I don't want to be part of that study to see how many percent of humans it kills. To me, I got to ask, why are we doing this, or why is BU doing this research? We just came from this pandemic, which was probably caused by a virus that came out of a lab. Let's let the scientific dust settle and give us time to reflect upon that before we actually start fiddling again with the virus in the lab. I don't know. That just seems like common sense. Let's pump the brakes on the whole thing. We do not have a compelling, immediate reason to do this type of research. The virus is effectively faded. You get the argument. Now, should things change and the risk profile and the cost and benefit, okay, I'm open to suggestion. If we have another, but I don't see that now. And it does seem as if BU is kind of, how should we say this, playing the bureaucratic game as well. We didn't have to tell you because you didn't directly fund it. And since we didn't kill all the mice, we didn't actually do it. Well, we tried to do it, but we weren't. They're playing two sides of the fence. And For those of you not from Boston, Massachusetts, you might not remember that when the lab was first sited there was a lot of controversy about what might go on at that lab, as there should be, and there was a lot of local opposition to it. Anyways, in the comments, you tell me. I'm John F. Gately. You can call me Jack from Plymouth, America's hometown, Massachusetts, behind enemy lines. Have a great day.